Hello and welcome back to Beyond Networks, the evolution of living systems. Today we're going to wrap up this module on dynamical systems theory and its tools by looking at historical theories of biological form. Before we go into that, just a quick repetition. We've collected this cart full of tools and pieces that we can puzzle together now to apply it to describe the study of the dynamics of morphogenetic processes. Okay, and these tools are, I told you in the beginning, in the first lecture, that dynamic systems are what they do. They're defined by their flow, which consists of the totality of all possible systems trajectories. This flow depends on the geometry or the topology of the phase space. The difference is the topology is just the qualitative features, which is, of this space, which is characterized by the phase portrait of the system that tells you how many attractors there are and how they sort of relate to each other. So the topology of phase space determines the position of attractors and their basins of attraction with respect to one another. And this arrangement, that is the topology of the phase space. This arrangement determines what the system can do, its dynamical repertoire, okay? Attractors are created and annihilated by bifurcations if you change the parameters of the system and the bifurcation set. So those lines in parameter space that we were looking at last time determine the probability of phenotypic transitions. And the last thing I was telling you is that we cannot assume that the system is in steady state. So it is not enough just to look at the attractors and how they're related to each other, but we also have to pay attention to the shape of the trajectories themselves, the transient behavior of the system before it reaches an attractor. And I showed you that there are tools that you can develop to also classify different types of such behavior in dynamical systems. So this gives us a toolkit with which we can describe, but also classify different types of developmental processes. And so we can ask the question now, armed with this toolkit, we can revisit this sort of statement with which Darwin ends The Origin of the Species after the first edition, so only from the second edition onwards. He has this beautiful last paragraph of the book, which I'm going to read to you. He says, "There is grandeur in this view of life, his evolutionary theory, with its several powers, having been originally breathed into a few forms or into one. And that, whilst this planet has gone cycling on, according to the fixed law of gravity, from so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved." So do we really have endless forms most beautiful? Or are there rules, regularities? Are there even laws that govern biological form? Could we use the characterization and classification of morphogenetic processes to come up with a general theory of form in biology? That question has been asked for centuries. There's a century-long debate in what we consider today evolutionary developmental biology between structuralists who believe that there are regularities, law-like regularities in how biological form is produced, and functionalists or adaptationists who think variation is more or less random and evolutionary dynamics is entirely determined by natural selection. And this goes way back. So the first person who wanted to come up with a systematic theory of biological form was, quite surprisingly, Johann Wolfgang von Goethe. He considered himself a poet only after the fact that he considered himself a scientist. And so he was studying different shapes of plants and was claiming that all of those shapes were variation, transformations of an archetypal Urpflanze, the archetypal plant. And he worked out a systematic phenomenological theory about this, with lots of drawings and transformation of shapes into one another, not an evolutionary theory. Okay, so this is important. Come back to that. But probably the most famous attempt at coming up with a general theory form was a book published in 1917 by Wentworth D'Arcy Thompson. It's called On Growth and Form, and it's a very famous book. It's basically sort of an early attempt by someone who wants to apply general mathematical and physical laws to biological shapes. So here's an example, very famous, of the variations of fish shapes. So he's making accurate drawings, anatomical drawings of different fishes, overlaying them with a grid, basically saying that all of those shapes are a fine transformation, so sort of rubber sheet transformation of this sort of average. This is a typical fish here. And if you sort of distort it in all of these different ways, you get the proportions of these other fish. So there's variation on a theme. I love this portrait of D'Arcy Thompson by Darren McFarlane, who uses one of his affine transformations here, one from the fishes, probably something like this, on D'Arcy Thompson himself. Here's the book cover. And apart from fishes and transformations like that, he compared cells to bubbles in a foam, or bones, vertebrate bones, to bridge girders and other elements of bridge construction, engineering. It's a very influential and inspirational book, but it had one big shortcoming. And I'm presenting you a quote by my own master supervisor, Brian Goodwin, that brings this criticism that he had of D'Arcy Thompson to a point. Goodwin writes, what is required in addition to what D'Arcy Thompson is presenting with these rubber sheets is an understanding of the organizational principles, we've heard that before in this lecture, that defines space-time order in the system under study, together with initial and boundary conditions. So that is how we specify a model. This dynamic model that he wants, that these are organizational principles, models based on those organizational principles, these are known as field theories. In biology, these principles are described as the properties of morphogenetic fields. We'll go into that in a second, which D'Arcy Thompson described in terms of energetic interactions. So the problem with D'Arcy Thompson's theory was that it was a static theory. It wasn't dynamic. It didn't consider the generative processes underlying these shapes. And these generative processes, according to Brian and a theory that he founded called process structuralism, what drives these processes are not gene regulatory networks like we think today, but what he called morphogenetic fields. This term was the foundation of classical embryology. So he didn't invent this term, but he took it from classical embryology. Just like we believe that genes are fundamental to development today, classical embryology thought that fields were the basic elements of development. And he describes these fields as structures describing the state of the whole organism. They undergo systematic transformation, they're dynamic, which conform to constraints defined by selection rules. This is a bit difficult. So these constraints are the rules by which development unfolds. And by selection rules, he doesn't mean evolution at all. He means that there are different possible dynamical behaviors that could be happening during development, but there are rules that guide which one of those sort of pre-given sets of rules are going to be implemented. And the whole process is in broad aspects self-regulating. Okay, so these fields are like magnetic fields. They can sort of fuse and merge and split and regulate themselves. So that's directly borrowed. The idea of a field is directly borrowed from physics and fields like electromagnetism. So these fields have global order and they have regulative capacities, but they're sort of mysterious. We'll look into what such a field is, specific examples in the next module. I'll give you some quick examples here though, but not in detail. So you can understand the morphogenetic field as a sort of a complex self-organizing process. And Goodwin thought that the genes were just setting the parameters of those fields. For example, you have a process that generates this beautiful twirled marine snail shell here. And there are two species of snail shells, fossil species that are found in Belgium in this case, and they look quite similar, but they are have a different handedness. One is left turning, the other one is dextro. So basically, there's the same morphogenetic field at work here, but there's a gene, and by now we know that there's a genetic switch that determines the chirality of the shell. Okay, so the field is what makes the shell look like a shell. It's the growth, the generative process that makes the shell look like a shell, and the genes choose whether the shell will be left-handed or right-handed. Okay, so this gives you a general intuition of what is meant by a morphogenetic field. Goodwin made a lot of very abstract models of fields and deliberately focused on systems that were not influenced particularly by genetic programs, if you want, or genetic influences more generally. One of these examples is a beautiful unicellular algae that grows in the Mediterranean and elsewhere. It's called Acetabularia acetabulum, or the mermaid's wineglass. It is several centimeters high. It's a single cell that consists of a complex morphology with a rhizoid. The nucleus is down here, a stalk. Then it forms these transient whorls and a mature cap. That's one single cell. And what Goodwin did with his collaborators, Trainer and Briere, is make a three-dimensional model of how this stalk grows, whorls, and forms the cap. So you have several symmetry breaking events that happen because the cell is interacting with the cell wall. There are different sort of concentrations of calcium involved that make the cell wall and the cytoplasm more or less elastic, and there's several feedback involved. It's a complicated model. You don't have to pay attention to the details at all. The bottom line is that these complex feedbacks between the cell wall and the cell form, self-organize into the specific shape, and then under specific circumstances form these sort of whorls that then form the mature cap. Okay, so two things here. There's a stereotypical sequence of these events that even happens if you cut the stalk off. So the nucleus is gone, but this process can self-organize for several hours before it runs out of steam. The other point that is very important is that you need those whorls. They have no apparent function, but apparently you cannot form the cap without the whorls. In the model, it always formed these whorls first before it formed the cap. So these are what you could call generic or sort of robust features of development that don't depend on the details of a genetic program. That was the point. So there's some sort of organizing, holistic organizing field here, morphogenetic field that shapes the morphology of this algae. And if you understand those fields, then you understand the geometry of those developmental trajectories that represent the generative processes that form the shape. So yet again, organization of matter together with these rules of directed asymmetric time-dependent transformation give you the generative principles that underlie morphogenetic fields. Goodwin was a student of Waddington, so it's not by chance that he's using Waddington's metaphor here. And what he does is he mathematizes Waddington's idea of a diachronic biology in terms of these morphogenetic fields. So this is a systematic theory of biological form, and what Goodwin believed is that these sets of rules that underlie the generative processes, these generative principles, they are universal. Okay, so he cited the Russian biologist Nikolai Vavilov, who worked in the 1920s, by saying that he had suggested that there is some sort of like a periodic table of elements for developmental shapes. So there's only a very limited number of different, qualitatively different developmental processes that are possible. And the aim of this theory was to catalog those and also catalog the possible transformation between these different processes, which would lead to what the famous structuralist biologist in the early 19th century, Geoffroy Saint-Hilaire, called a rational taxonomy of biological forms. So this is Goethe's project again. Brian was also heavily influenced by Goethe. This idea that you can relate biological forms to each other via the generative principles that form them. This is a taxonomy that has nothing to do with phylogeny or common ancestors. It's a rational, just like a periodic table. It is a rational sort of arrangement, systematic classification and arrangement of possible shapes. So not endless forms most beautiful, but only a limited set of forms that can be related to each other. There is a different way, slightly different way to look at this. And for that, we have to turn back to this idea of the genotype-phenotype map, and that developmental processes represent such a mapping between genotype and phenotype space. Let's go back to Waddington, where everything always begins here. Waddington in 1968, in one of his later publications, has this beautiful picture of a genotype-phenotype mapping. The genotype space is here, phenotype here. He even maps it onto fitness. But look, in the middle here is what he calls epigenetic space. This is where his landscape lives, the epigenotype of the organism. And it mediates between the genotype and the phenotype. And we can use dynamical systems theory to fill this epigenetic space with meaning. So basically, we have a genotype space, and the genes implement different network behavior, dynamic behaviors here. For example, if the toggle switch, we have these two phase portraits. Again, here's a bistable and a tristable toggle switch. And through these dynamical regimes of the developmental processes systems, you get to a phenotype space. So in this case, you start with a bit more green and then go up to the green, the trait value, high levels of the green protein, while here the green protein goes up for a while and then goes down again. So very different qualitative developmental trajectories. So what is in between the genotype space and the phenotype space? It's Waddington's landscape, and if you make that precise, it's the potential landscape of the dynamical system that governs development. And so apart from Goodwin, there were a number of other people who took this idea very serious in the 60s, the 70s, and then the early 80s. And I want to point out three people here in particular that could be grouped into a similar schools of thought, although they worked independently. So the first is Pere Alberg and George Oster. Pere Alberg, the evolutionary developmental biologist, Catalan evolutionary developmental biologist, very eccentric, very famous. We've encountered him already when we looked at the salamander limbs in last lecture. George Oster is a theoretical computational biologist and modeler. And Rupert Riedel, of course, was in Vienna, the head of and the founder of the theoretical biology department, and a pioneer in what he called evolutionary systems biology. And these people were wondering about a curious fact of shapes, of phenotypes in nature, and that is they come in discrete packages. Alberg and Oster call these morphological themes. Riedel calls them morphotypes. So why are there no intermediate forms? You can have several explanations for this, right? So one is it may be that natural selection favors discrete forms, but that doesn't really make sense in all cases. It could be history. So there are missing phenotypes in between those islands of form that we see because they simply have not had time to evolve yet. And that is Goodwin's answer, and the other structuralists, Alberg, Oster, and Riedel, there could be internal developmental constraints. And what we're actually seeing here, these themes are there because they represent the outcomes of complex developmental processes, of fields, of generative processes in Goodwin's framework, while in a dynamical systems framework, you could say these are these islands of form that we see are formed by stability domains, basins of attraction that systems tend to fall in and that dominate other modes of developing. So the discontinuity between these forms is the consequence of a threshold effect. It's a threshold phenomenon produced by the underlying bifurcations in the system, which we have covered in last lecture. Okay, so Oster and Alberg elaborate this with a specific example. And this example is beautiful. It's the formation of skin structures in different animals. And you can see here, if you look at the center, all of these structures start the same way. So there is an epidermal placoat that forms a thickening of the epidermis on top of a mesenchyme, and underneath is a dermal papilla, a mesenchymal structure, a condensation that seems to be triggering the formation of the placoat. Okay, and this happens in all of these cases, but then if you go along different branches here, the developmental pathway diverges. So this is still research that's very relevant today. Just very recently, 4 years ago, the group of Milinkovic in Geneva published molecular evidence that these placoats in all kinds of vertebrates for all kinds of structures are indeed homologous. Now, this homologous structure forms vastly different shapes: feathers, scales, glands, spikes, teeth, horns, hair. Okay, and the crucial difference, look at the different branches, is that in some cases the placoat will bulge outward, and in some cases it will bulge inward. Okay, if you form a feather or hair, it goes inward. If you form a scale, it bulges outward. The same for spikes or horns, but teeth are more like feathers in that they bulge inward, surprisingly. Okay, so you have this branching point here. And this crucial point where the tissue decides to go up or down, this is one of these symmetry breaking events that's very important for the formation of body shapes. Okay, so how does this decision happen? Okay, the outcomes are vastly different, but the difference that causes this tissue to bulge outwards or invaginate inwards is very small. You can model this with a metal rod that's attached to a wall, and you exert pressure. Okay, from the right hand side you have force, and so the rod eventually, above a critical threshold of that force, will buckle. Okay, if you push right in the middle and the rod is completely symmetrical, it'll buckle up or down completely randomly. But by very small differences that you induce here, a little asymmetry, or even by just applying the force a little off-center, you can bias this process towards one or the other direction. And so the idea is very similar to this idea by Goodwin that a gene causes the snail shell to curl left or rightwards. The idea here is that a gene sets the parameter, the offset, if you will, of this force. This is very metaphorical, but this is the principle that biases this process to go outwards or inward. And you can form really stable responses this way. So what's happening here is you can draw this in an abstract space. Okay, so this space is neither parameter space nor phase space. It's both. Okay, so you vary different parameters, like the offset E here on the x-axis. This is the strength of the force. So this is not a parameter. This is a variable of the system. And then up here you have the material properties. The offset, sorry, this is also variable. This is the amount of buckling that you see. So it goes upwards or it goes downwards. And if you increase the force, first you can see that it sort of buckles, you know, if you're left or right, it buckles left or right, no problem. But then as you increase the force, there's this critical point here where something funny happens. So as soon as the rod has buckled, it's really hard to push it back to the other direction. So what you get is this sort of folded shape here. And this is called a cusp bifurcation. So if you abstract this system into this weird space, configuration space, you can draw the shape of its behavior. Okay, so the theory that Alberg and Oster suggest here is that evolution is driven by such bifurcation events. And now we're going to get really crazy for about 15 minutes. You don't have to remember or understand the details of what I'm going to do now, but there was a crazy mathematician Fields Medal winner, that's like the Nobel Prize for mathematicians, René Thom, who happened to read Waddington's Strategy of the Genes. You should really read that book. And he set himself a life goal, and that was to mathematize Waddington's metaphor of this landscape. He wanted to have an analytical, rigorous mathematical theory of how these landscapes look like. So no experiments, everything top down, general theory. And he started with these sort of shapes, very simple shapes in configuration space. Here, the surface again represents this, the steady state of the system. It buckles up or it buckles down, remember. And you have two folds here. These are important. So if you fall over this edge here, you fall down. And then you have this point where the fold starts to come into existence. It's called a cusp because if you project it down to parameter space, it has this sort of shape here. So this sort of phenomenon in this sort of shape in parameter space, this sort of fold, represents a process called hysteresis. It happens also in that bistable genetic switch I was telling you about. So if you switch the system into a stable state, high cruple, low giant, or high giant, high cruple, it's much more difficult to flip it than if you start somewhere with equal concentration of both factors. The rod is easy to buckle if it's straight, but once it's buckled and the force is strong enough, it's really hard to buckle it the other way. This effect is called hysteresis, and it makes the system, the state of the system, history dependent. So where you start is very important for the ultimate outcome of the behavior. Again, so you cannot predict the behavior of the system from its structure alone. You have to take into account the history, the initial conditions of the system, very carefully here. Okay, Thom then went on and wrote a crazy book. So he used a mathematical theory called algebraic topology to look at the possible shapes of these bifurcations for systems with 2 or 3 variables and parameters. So he went up to 5 dimensions, 5-dimensional space, and he found something really, really surprising. Okay, first of all, he found beautiful structures. This one is the next one up from the cusp that I was showing you before. It's called the Swallowtail, and you have these beautiful intersecting sort of surfaces. Don't worry too much about what the mathematical details are here. Just know that this describes qualitatively different ways of undergoing a bifurcation, which would then result in qualitatively different ways of breaking symmetry during the development. It's a very abstract theory. You don't need to understand the details. So you can imagine this sort of shape, like this shape in this picture between the 2 hills here, the Paps of Jura in Scotland. And this is from an absolutely wonderfully weird book that explains Thom's theory based on nude photography. So you can look at lots of nude people, men and women, and he'll explain to you not just the Paps of Jura, that's up to your imagination, but human shapes that contain such different catastrophes. It's a wonder, it's a little weird, but it's a wonderful way, a memorable way to explain this theory in a very non-technical manner. And he has beautiful drawings in there, but before I have to present the one result that you have to remember here. Don't worry about the mathematics, okay? But the fact here that you need to remember is that this table represents systems that have up to 4 parameters and 2 variables, so 6-dimensional spaces. And what Thom found is that in these 6-dimensional, in anything that has any configuration space that has less than 6 dimensions, you can only get 7 qualitatively different types of bifurcation. The cool thing is he could prove this mathematically. Unfortunately, only for a subset of dynamical systems. We'll come back to that in a minute. But this is an important result. Why? Because it's a mathematical proof of principle that there are not endless forms most beautiful. There are only very limited ways in undergoing symmetry breaking bifurcations. So there are only 7 different ways, basically, more or less, of undergoing development. Qualitatively different ways. Of course, there's endless variations on this, but basically, there is hope that we can get this sort of periodic table of elements for developmental processes, because there's only 6 possibilities. He gave them fancy names. So the fold, the cusp, the swallowtail, the butterfly, they're cuspoids, and then more complicated structures that are called umbilics. I just can't help myself but quickly introduce those. Here's the fold, the cusp, the swallowtail. We found there's a butterfly that is slightly more complicated. The umbilics are really crazy. So look at this shape here. It's a weird folded shape of 2 surfaces that cut through each other below this line here, right? So they sort of cut through each other. And you can construct this shape, it's called a Whitney umbrella, by cutting through a sheet of paper like this, and then folding the sheets in 2 different directions, and gluing them onto the sheet. So here, you have to imagine that these 2, so this goes through the other surface here, right? Okay. So what am I telling you here? You can construct 2 of the umbilics out of this paper cut here. So if you then bend this umbrella upwards, you get what's called an elliptic umbilic. Beautiful shape like this. And if you bend it downwards, you get what's called a hyperbolic umbilic. There's one more figure here, which is called a parabolic umbilic that you cannot even visualize in 3 dimensions. So it's really hard to see it. So here's a series of graph that is maybe giving you an intuition of how it looks like. It's not working for me. But the amazing thing is, this is not just amazingly beautiful and elegant mathematics. It's an amazing insight. Only 7 different, qualitatively different ways in which you can do a bifurcation in systems that have less than 6 dimensions. If you imagine that you're describing events that involve more than 6 factors, they're getting increasingly unlikely. So these are the most likely events to happen in nature. And this was work that inspired not just scientists, but Salvador Dali, for example, was impressed by Thom's beautiful work, and he set out to draw all of Thom's high-dimensional attractor structures. And unfortunately, the swallow's tail that you may have noticed in the back of me lecturing sometimes in a wall, unfortunately not today, the light doesn't allow me to film there today, but this was unfortunately Dali's last painting. He never got to paint the rest of them. Okay, so this is crazy stuff, and it brings us right back to the beginning when we started talking about process philosophy. Here's Thom. I can't help myself. You don't need to remember this. It's just beautiful. That's why I'm telling you this. He's writing that what he's doing really is just formalizing the ideas of Heraclitus, who said everything flows. Remember him? The father of everything is strife. Everything is sort of a dialectic process. And so Thom writes, the pre-Socratic flavor of the qualitative dynamics described here will be quite obvious. Nothing is obvious in this book, really. It's a really hard book to read. But if I have quoted the aphorisms of Heraclitus at the beginning of some chapters, the reason is that nothing else could be better adapted to this type of study. In fact, all the basic intuitive ideas of morphogenesis can be found in Heraclitus, these little fragments that we have of this philosopher. All that I have done is to place these in a geometric and dynamic framework that will make them someday accessible to qualitative analysis. The solemn, unadorned words, like those of the Sibyl, that have sounded without faltering throughout the centuries, deserve this distant echo. So Heraclitus should be weeping. He's called the weeping philosopher. He should be weeping no more, because Thom has built a beautiful mathematical theory around his ideas. A theory of dynamic forms that maps back onto Waddington's landscape, in that the shape of the creodes, the trajectories in the landscape, they are molded by catastrophes, which are structurally stable bifurcations that give you these sort of branching paths of the landscape. So if we have simulated, with the toggle switch, we have simulated Waddington's landscape in a very crude sense, Thom has come up with a beautiful mathematical theory of telling us what is possible in this landscape. There's only one drawback, of course, and that is it's totally abstract. I was telling you that it only applies to a certain subset of dynamical systems. So these gene networks or any regulatory network, unfortunately, don't belong to that type of system. So it doesn't really apply. Thom, in the whole second half of his book, goes completely off the edge and tries to explain, to generalize his insights. But it's a slightly weird experience to read that. But why is it still useful if it's completely abstract like that? It's because Thom gets some really interesting points right, okay? And so one is morphogenesis is a geometrical problem that cannot be tackled by biochemistry alone. Duh. More precisely, it's a problem of the geometry of configuration space, or the topology of configuration space. And if you think about it, what we've done here is we've classified developmental processes according to topological feature of an abstract space. Not network spaces, but this configuration space I was telling you about. You can show philosophically and mathematically that the two are equivalent. So what we have here is a form of topological explanation, not mechanistic. There's nothing mechanistic about these configuration space shapes. They're very abstract. They don't connect to any causal structures, any causal explanations yet. But by themselves, they are exactly like these global network analyses by Barabashi and other people that infer certain types of typical behavior, anti-fragility, robustness, modularity from general network topological features. So in nature, this is a topological theory of now, instead of sort of the robustness of a system, like in the network case, of the, it's also about robustness, which is called structural stability in this case. But what's really important in our context is that there's only a limited amount of what can happen during development, the types of bifurcations that can drive development. And it is possible to catalog and classify all possible catastrophes. So these bifurcations. Did I introduce that term? These bifurcations were called catastrophes. I should have introduced that more clearly. So systematic classification of a small number of elementary catastrophes, bifurcations, that's a solid, profound, and elementary insight based on a rigorous mathematical proof. So it should be possible, we think, to classify different types of morphogenetic processes in the real world. The problem is that the top-down theory doesn't really connect to anything. So I'm going to end this lecture in this module on a rather long quote from one of my own papers. It's not a very gracious thing to do, but I think it's very sort of appropriate here. So we propose, the authors of this paper, me and my two colleagues here, we propose something resembling a series of targeted expeditions into the vast unknown territory of configurations. So the general theory is too abstract, it doesn't connect. But we can simulate, like we did for the toggle switch, these landscapes in configuration space for specific biological systems. Although we cannot gain a complete overview of the landscape yet, no general theory, surely we could gain a lot of exciting new insights by just having a look around, exploring this topic. This seems a sensible approach in light of the fact that nobody has ever explored this territory before. How do you explore new territory? You don't draw a systematic map, you just go and have a look. We call this the natural history of configuration space. Instead of Darwin in a beagle, we will go into this abstract space in the imagination, in analogy to the real-world expeditions that have led humanity to discover new countries, continents, and even new worlds beyond our own planet. Such expeditions always preceded the systematic mapping of the newfound frontier, not only in natural history proper, but also in many other branches of biology and science. So with that, I'll end this really, really monstrous module. You've covered dynamical systems theory, something that takes several semesters of lectures, usually in a mathematical curriculum. We've covered this ultra fast. If you don't get the details, that wasn't the point. The point was just to give you an insight of what the concepts are of this theory and how they describe different processes and allow us to classify them, okay? So we have now a topological explanation of how different developmental processes qualitatively differ from each other. What we need to do, of course, is now to connect this back to the real world, to look at how we can explain these phenomena mechanistically, to connect them back to the gene networks and the mechanisms that drive these sort of processes. So in the next module, we'll look, first of all, we'll need to look at causality and what that actually means. And then we will try and apply the principles here. In the spirit of this humongous quote here, we will try to apply the tools we've developed in this module to the study of real-world developmental systems, and we'll use the gap gene system again as an example. I hope you're still with me on this. This has been sort of the intellectual hump, I think. There may be another, it may be slightly camel shaped when we come to the theory, organizational theory of organisms, but this was the most technical part of the lecture, I assure you. And I hope you'll tune in again next time when we'll talk about causality in complex systems. Thank you very much for listening. Goodbye now.